My name is Megan Mitchie, and I am the first author of our paper entitled Cyanin Conformational Restraint in the Far Red Range, which I worked on while I was a postbac at the NIH in Dr. Martin Schnerman's lab. An important goal in optical imaging is to make tools that better visualize structures in complex living environments. Fluorescence imaging experiments have been very useful in this context. Fluorescence-based experiments use molecules that are activated by exposure to a certain wavelength of light. Once activated, the molecules, or fluorophores, emit light of different longer wavelengths that allow researchers to locate structures that are labeled with a specific dye. These tools are important for a better understanding in both experimental and clinical applications, such as fluorescence-guided surgery. While these techniques work well for cells and tissues examined outside of the body, visualizing fluorescence in living organisms is difficult because light from most existing fluorophores does not pass through tissue well. Most currently available fluorescent dyes work in the green and blue range of the visible light spectrum. Our lab has been focused on fluorophores that emit longer wavelengths of light in the far red and near infrared range, longer than 650 nanometers. We are interested in these longer wavelengths because they are better able to penetrate through tissue, which is something everyone has seen when you hold a flashlight to your hand and the red light passes through it. However, most fluorophores that emit light in these wavelengths are not bright enough for biological applications. Figuring out how to make molecules that are both bright enough and emit long wavelengths of light has been a long-standing puzzle for chemists. Most of our work has been on a class of fluorescent probes called the cyanins, sometimes called the Cy dyes, which have strong light-absorbing properties. However, as fluorophores, they are not very bright, which limits their use for addressing many important problems. Our lab speculated that the reason cyanins are not very bright is because these molecules rotate after they absorb light. We made the hypothesis that restricting their ability to rotate by attaching a rigid structure would make them brighter. Using tricks from modern organic chemistry, we built a system of chemical rings and attached them to the far red Cy5 fluorophores to form what we call conformationally restrained Cy5, or Cy5b for short. With this novel synthesis complete, we were delighted to discover that the addition of these new ring structures made the fluorophores over 400% brighter. In addition, these molecules also have a longer fluorescence lifetime, which will be useful for visualizing time-dependent changes in tissues and cells. In addition to being brighter, conformationally restrained Cy5s have an improved ability to turn from on to off, which makes their signal easier to distinguish from the background. These new properties prove to be quite useful for super-resolution light microscopy, SMLM, imaging applications. In SMLM, molecules in a tissue sample, such as tumor cell surface markers, are fluorescently labeled and turned on to a bright state one at a time, as is shown in this movie. This allows researchers to count them and determine their location with nanometer precision. This approach has been used to localize structures both inside and outside of cells, as is seen in this example, imaging actin with these new dyes. Going forward, these newly synthesized Cy5 far red fluorescent probes are brighter and more stable than the currently available counterparts, which means they could be used for imaging live tissue over long time frames. These promising properties point to several applications, such as tracking tumor cells in live tissues, which could lead to a better understanding of how cancer behaves in a living system.
U.S. Department of Health and Human Services, National Institutes of Health, National Cancer Institute, cancer.gov, 1-800-4CANCER, produced October 2019.